Hi, I'm Noah. Did you know that while you and I can't choose to change our DNA can choose to change itself? How does it do this?
Jumping genes! Let's start with what jumping genes are. They are DNA sequences that are capable of moving from one location in a cell's DNA to another, and are also known as transposons, or transposable elements. In 1944, Barbara McClintock discovered their existence through her study of genetics in maize or corn. Finally, while there are many different types of jumping genes, they can be divided into two main categories: DNA transposons and retrotransposons. What are DNA transposons? They produce or encode an enzyme named transposase that essentially allows them to cut and paste themselves from one place in the genome to another. But how does this work? Well, at the ends of every DNA transposon are a pair of terminal inverted repeats, which are complementary to each other. For example, if at the start of a transposon there's a sequence ACG, at the end of the transposon there will be the sequence TGC. These terminal inverted repeats show the transposase where the transposon begins or ends, and therefore where to cut it out. However, while jumping genes make up at least 45% of the human genome, DNA transposons themselves are only about 2 to 3% of it. The rest are retrotransposons. Now let's talk about these retrotransposons. They're unlike DNA transposons because they leave the original transposon in place and move by copying and pasting their sequence throughout the genome. So how do they work? First, the transposon undergoes a process called transcription in order to create an RNA intermediate, or RNA copy, of the original genetic sequence. However, the RNA intermediate will actually be a complementary sequence to the original gene, not an exact copy. Because of this, the RNA intermediate is then reverse transcribed using what are called reverse transcriptase enzymes back into a DNA sequence that is the exact copy of the original transposon. Finally, this DNA clone is inserted at any location in the genome. So what do jumping genes actually do besides for jump? When they land inside a gene, they can cause genetic mutations. However, their jumps can actually be beneficial. They can drive evolution. In fact, researchers have found that about 35 million years ago, a gene jumped in spider monkey's ancestor and led to the imperfect duplication of a gene named CHMP3. This imperfect copy, known as retro-CHMP3, might actually inhibit HIV from spreading. They believe that they can design strategies that mimic retro-CHMP3 activity in human cells and thus block HIV replication. Jumping genes might hold the cure to HIV and other viruses.